Parkinson's disease, or PD, is the second most common neurodegenerative disorder affecting the brain and other parts of the body. It is commonly seen as a movement disorder due to its prominent motor symptoms such as slow movement, muscle rigidity, and rest tremor. However, there are also many non-motor symptoms often observed in Parkinson's patients, such as loss of smell, digestive problems, and sleep disorders. Some of the most common are digestive problems, including constipation, bloating, and abdominal pain. These problems may be present years before the onset of motor symptoms. In other words, they may indicate the early stages of Parkinson's and provide an early warning system for patients and healthcare providers. While Parkinson's disease is considered a brain problem, mounting evidence suggests it may actually begin in the digestive system. Therefore, it is more important than ever to uncover and understand the relationship between the gut and the brain. The enteric nervous system, which controls the function of the gut, communicates with the brain via the vagus nerve. This nerve forms the gut-brain axis, a two-way highway where messages are transmitted in both directions. The gut end of this nerve appears to be the origin of a pathogenic protein that can propagate to the brain, possibly causing a variety of neurodegenerative diseases, including Parkinson's disease. While it is yet unclear what triggers this pathogenic protein to develop, several factors have been identified. Here is one possible scenario. The gut is colonized with a massive community of microbes, some of which may produce harmful substances that can enter the cells lining the gut or pass through the leaky barrier. Over time, these may trigger a pathologic change to surrounding neurons in the enteric nervous system. Inside the neuron, a native protein called alpha-synuclein can misfold due to several factors, including the harmful runoff from the gut microbes. These misfolded proteins rapidly aggregate into small repeated units called oligomers, which continue to elongate and assemble into long fibrils. Once formed, the fibrils can spread and be transmitted from neuron to neuron. Once inside an adjacent neuron, the fibrils act as templates to seed misfolding of normal alpha-synuclein proteins, causing the entire process to repeat. The fibrils are toxic to neurons and eventually cause parthanatose, a type of cell death. After repeated steps of misfolding, aggregation, and transmission, more and more neurons along the vagus nerve accumulate the toxic fibrils. With time, the pathology propagates to reach the brain, ultimately causing the symptoms associated with Parkinson's disease. Although the cause of the initial misfolding in the gut remains unclear, the gut-to-brain spread of the pathogenic protein, as well as the early onset of digestive problems, suggests that Parkinson's may start in the gut. Further research may lead to early diagnosis of Parkinson's disease and pave the way for effective early interventions that target the gut instead of the brain.